Hi everyone, this is Mr. Sinti, and this particular lesson is on the nucleus, and it's also on ribosomes. As we're going to see in this lesson that the nucleus, which is the most prominent organelle in the cell, most conspicuous, most easily visible, you might know it. Nucleus itself, we talk about the nucleus in terms of like the atom as well. Nucleus meaning that, like the central area, or you could say that the library is the nucleus of the skull. It's basically the central, most important area, and I think you know it somewhere in your past as being the place where DNA is stored. And so eukaryotic cells possess this membrane-bound organelle that's rather large called the nucleus. You could see it here. Eukaryotic cells have this, but a prokaryotic cell, remember, doesn't possess it, so it's one of the defining distinctions. And before I get into the notes, talk about this, I want to do a little bit of a background on this, and so let's see if I can. I'm going to go here and draw a couple of things about a typical cell. Like, let's say if I had a cell like this, and what I'm drawing here is the outer membrane, or the plasma membrane, and then it's sort of like the nucleus is a cell within a cell. It's also surrounded by a membrane itself, and so it's not considered to be the plasma membrane like the outer cell membrane, but it's considered to be the nuclear membrane, or if you will, nuclear envelope. Sometimes that's kind of a cool thing. And so what I want to say about that is inside the nucleus is a very important molecule that you might know is present. It's a molecule that's made up in many, many strands like this. And all these strands inside are of this polymer called DNA, or polynucleotide. So I'm just going to label it like this, DNA. You're like, what's that DNA doing? Well, it's the recipe of how to make proteins. But proteins are made in little tiny structures, the organelles called ribosomes. They're the most numerous and smallest of all organelles. Now, I'm not drawing them all in. If I were to draw them in, there'd be millions in the cell. And so what's kind of interesting is there's a relationship between the nucleus and ribosomes, because ribosomes are where proteins are made, but it's the DNA that contains the recipe of how to make it. And it's like, okay, how's that work? Well, as you may know, that all cells are surrounded by blood. So there's a blood vessel next to every cell, called a capillary. And so in the blood is red blood cells. Let me draw in a red blood cell, white blood cell. And so also in the plasma of the blood, the fluid in the blood, there's little tiny amino acids. And it's like, well, how did those amino acids get there? These 20 different amino acids come from the food that we eat. So we've eaten some kind of protein. And those amino acids diffuse into the cell through a protein, facilitated diffusion, because they're polar molecules. And so when they get into the cell, they go to the ribosome, and the ribosome assembles amino acids into proteins. And that's basically just a dehydration synthesis where peptides are linked together, and for example, they're linked together like this into long chains, and then those fold into unique structures that we know as polypeptides or proteins. This is obviously very large, not drawn to scale, but there's a protein. And it's like, okay, there we are. But what's interesting is the cell makes many different kinds of protein. And if you remember our discussion of protein, what's the difference between one protein and another protein? It's the sequence of amino acid. It's sort of like, what's the difference between your name and someone else's name? It's the letters and the sequence, or your phone number and someone else's phone number. It's the numbers and the sequence. So likewise in a protein, it's the amino acid sequence which is critical, because then that folds into different shapes. The different shapes add different functionality to the protein. So my point is, the ribosomes are sort of like a cooking island in your kitchen. So in other words, like the ingredients or amino acids are assembled on these cooking islands into whatever the particular protein it is that you're interested in. But the cooking island can't do anything in and of itself, and the ingredients aren't going to do anything. What you need is a recipe. And the recipe for protein is the amino acid sequence. And so, like, what is that? How can you get a recipe? So the recipe is over here in the nucleus. It's in the DNA. Now, I'm not, though I could get into the detail of that. We will discuss it later in the second semester. But I'll say that the DNA contains on it the recipe for all of the different kinds of protein. It's sort of like a cookbook, that like each chapter has a different recipe. And this is the recipe for every single protein in the whole body. But if you're going to be making this particular protein like that right here, like switch my analogy, say it like this is a restaurant, somebody comes in, they order some kind of pasta dish. Now, the chef isn't going to create every single recipe. Only that particular one is going to be made. So what's interesting is the DNA doesn't come out into the cytoplasm and say, all right, here's all of the recipes to make all the different proteins. But instead, it's more clever. First off, it can't leave the nucleus because it's too large. And so what it does is, you may know this somewhere from your past, but it can make a little copy of this one area for gene. It's called a gene. It's a segment of DNA. And that gene, right there, you've heard that term before, that gene or segment of DNA is the recipe for that particular protein. And so what happens is the cell makes a copy of it into another molecule called RNA, and it can make many copies of RNA. It's sort of like photocopies. You don't want to bring the whole book out. You just want that particular recipe. So it makes many photocopies because there's many millions of ribosomes. So these guys are small enough to leave the nucleus. And so the RNA comes out and it has the recipe to make these different proteins. And so the nucleus and the ribosomes are related because ribosomes make proteins. The nucleus is where the recipe to make the protein is located. But DNA itself doesn't do it. It's this sort of intermediate called RNA. And so if I could simply spell it out, it's DNA in the nucleus codes for this little messenger, sometimes called messenger RNA, which is like here. And it leaves the nucleus and it goes to a ribosome to make a protein. And so this sort of sequence of DNA, messenger RNA, and proteins are really important in biology. And so that's what I wanted to say about that. Let's go back to the slideshow in hand and see if we can get that to open up. And so here we have the nucleus. I'm going to sort of illustrate that. I wanted to actually not do this. Here we are. And so what I want to say about this is that the nucleus, you could see the nucleus here under the transmission electron microscope, and here's a cartoon drawing of it. It contains most of the cell's DNA. In fact, we've learned that mitochondria and chloroplasts contain a little bit of DNA as well, which is kind of unusual. But in the second semester, we're going to talk a little bit more about how that might have come to be. The fact that cells incorporated other cells early on in evolution, and so they became symbionts. And so that's kind of a cool idea. So there's some DNA in the mitochondria and chloroplast. First thing I want to say about the nucleus is that it's separated from the cytoplasm by its own membrane. I mentioned that before. And it happens to be a double membrane, not phospholipid bilayer, but double phospholipid bilayer. And so here's a picture of the nucleus, and what you can see here are there's visible pores. These are like actual openings to the nucleus. This is not the whole cell, this is just the nucleus. And it's like, well, what's going on with the pores? The pores are happening. Let's see, sorry about that, I don't want that to happen. Let's see if I can do this. Maybe if I click here. Okay, sorry about that. These pores, which are located here, need to be there because, as I mentioned before, DNA is sort of inside, so we can't see it. But what's coming out of the nucleus is this molecule called RNA. And it needs to come out because over here in the cytoplasm are the ribosomes and where proteins are made. And so, like, why do you need to have pores? Because can't something travel through a protein in the phospholipid bilayer? And in fact, nothing this large can get through a protein. And so let's take a look at that notion, if we can. The nuclear membrane, and here's a real close-up of the nucleus right here. The nucleus actually has pores. If you can see right here and right here, these are physical gaps where RNA is able to pass out of the nucleus. Here's some of the most powerful images that we have of the nucleus. It sort of looks like, I don't know, let's say it looks like craters of the moon. These little holes right here, and this is the pore itself. The pore is surrounded by a bunch of proteins. And so the only way that you can get a pore is when you have a double phospholipid bilayer. And so I think this next image will help you understand that. If you have one phospholipid bilayer, on occasion, you can have proteins embedded in it. We've talked about this. These proteins can allow the movement of some molecules in and out. So there's some proteins on the outside. But RNA is way too big, or proteins are way too big to pass through a protein embedded in the phospholipid bilayer. So if you had a protein like this, nothing would be able to pass through. And so you're thinking, why would two membranes be easier than one? You would think two membranes, nothing would be able to pass through. But in fact, two membranes makes it easier for large molecules to pass in and out of the nucleus. And why is that? Because at certain areas in the nuclear envelope, these proteins, difficult to draw, but these proteins can sort of coalesce in a particular area. And what can happen is they can form a real huge physical pore that will allow for RNA to exit the nucleus. And so a single membrane would be restrictive. A double membrane allows for large molecules to pass through, and that's important. And so I like this picture. Again, this is the transmission electron microscope, and you can see, let me sort of illustrate it the way I was doing it before. Like there's a protein that can be embedded in the membrane there, a protein there, protein there, protein there. But then when you get a bunch of proteins together, it forms a pore, as you can see here. Why is the pore important again? Because it switch colors. Say this is the inside of the cell, this is the outside of the cell. Say that this is your DNA that's really huge, and even this is too large to pass through a pore. But what it does is it makes a copy into RNA. Now RNA is too big to pass through the membrane, but it can leave through a nuclear pore, and so that's kind of critical, if you will. And so here's a picture of the nucleus, and I just want to focus in on the membrane or the nuclear envelope. And so let me read this. Where there's double membranes fused together, a large pore sort of materializes. And so as you can see here, there's one membrane and another membrane, and then a cluster of proteins together forming these sort of pore complex. And it's like, well, do we know that that actually exists or not? Like, I mean, can we see that? Some of our most powerful microscopes can see this. So here's the membrane of the nucleus. And then really close up, you can sort of looking down, see, these are kind of like those manholes on the street where they're like little places where you can like open it up and go through. And so let me sort of back off of this for a second and show you, I have like a little video clip of this. And so let me show you, here you can see a close-up of the nucleus, and you can see these like sort of pink structures as being the nuclear pore. Let me let it continue to play. This allows the passage of very large molecules in and out of the nucleus. So the nuclear membrane is not as discriminating as the plasma membrane because it's a double phospholipid bilayer, and so therefore it creates these pores where large things can pass through. And so like, if we were to travel into the nucleus, we would say, whoa, okay, these are all strands of DNA, all inside here. The DNA is being illustrated by these blue threads. And you're like, well, what's going on? These threads are actually wrapped around proteins that are in the nucleus called histone proteins, which we'll be talking about a little bit more during mitosis. And so the point being is there's so much DNA in there that it wouldn't be able to fit unless it was coiled up, sort of like yarn around some sort of plastic. And so as you can see here, the DNA is coiled up, and then what's going to happen is a segment of DNA needs to be read. And so this area right in here called a gene, this is a enzyme that's going to speed up that process. And so that little piece that went really is RNA. Now RNA, if we continue this, you might be able to predict that the RNA that's produced, it's destined to leave the nucleus. And so it's small enough, if you will, to be able to travel out of the nuclear pore. So let's go back over here to our slideshow, and let's see if we can do that. So what I want to say is that a term to introduce is the fact that when you look at the nucleus, it's sort of, when scientists first viewed this under the microscope, they saw this as a circular structure mainly. They saw it sort of standing out when they put dye onto a cell to be viewed under the microscope. And they saw like threads inside the nucleus, like we just saw in that video. And they thought, wow, what is that colored material inside the nucleus? And so the term, it's kind of an older term, but it's still used. What is that colored material? So the word chroma means color. So colored material, what is it? So it's DNA inside in the nucleus. And I was mentioning before, there's protein in there. And so let me change the color. So there's proteins in there as well. And so collectively, inside the nucleus, dramatic like there, inside the nucleus, here's the nucleus. What's inside the nucleus is chromatin. Now, this is a little foreshadowing to a future discussion, is that when the cell divides, let me go like this, here's a cell. When the cell divides into two cells like this, here's a little bit of an issue. The DNA needs to replicate itself, or else the new daughter cell is not going to have any DNA at all. And so one of the dilemmas that the cell has is that there's so much DNA in here and it's all kind of loose. It's like it's difficult to travel with this. And so in order to get away with it, what the cell does is that it takes its DNA and it coils it around these proteins like we saw in the video, but even more so. Coils and coils and coils until it forms like these really dense structures called chromosomes. Now there's a picture of a chromosome with the scanning electron microscope. Chromosomes form before the cell divides so that the DNA can easily travel. But you might be thinking the DNA is not going anywhere because there's a nuclear envelope. And so we'll talk about that later. The nuclear envelope disappears or disintegrates, doesn't disappear, disintegrates right before the cell divides. And chromosomes form right before the cell divides in a process called mitosis. You might be familiar with it. So I wanted to mention that before the cell divides, the chromatin does condense into these big structures called chromosomes. And so what's interesting is that different organisms have different amounts of DNA. And so these fibers can condense into what would be 11 chromosomes, 76 chromosomes. Our number happens to be 46 chromosomes. You might recall that somewhere in the past. 46 chromosomes contain all of our DNA, and then we move them in a process called mitosis before the cell divides. So that's kind of interesting. So the DNA, which is this double helix, contains in the structure of these nitrogen bases, which we'll talk about later, the recipe on how to make a protein. And remember, all of that is contained inside the nucleus. But you might be saying, okay, if this is the nucleus, what is all this dark structure? Like, what is that? This dark and dense region that stains even darker than the nucleus itself is called the nucleolus. So the dark region, nucleolus. And if you have a lot of nucleolus, it's called nucleoli. What is it? Well, to get into that discussion of nucleolus, I have to talk a little bit about ribosomes again. There's a connection there because ribosomes themselves are produced in part in the nucleus, and then they're released into the cytoplasm. And where they're produced in the nucleus is in this dark region called the nucleolus. And so let me back it off over here and see if I can come to my slideshow over here. Maybe if I come over here, perhaps. Let's be hopeful. So let's say that here's our cell, the plasma membrane, and here's the nuclear membrane. And I'm going to draw it like this because you now know it to be a double phospholipid bilayer. So let's go like that. So double. And I'll add a little detail. Let's say there's some proteins here like this, and a bunch of them here like that. These areas are called the nuclear pores right here. And it's like, okay. So what happens is, and this is real brief, but I'll try to be clear about it. So DNA is located here, and I said that DNA carries in it, with it, little segments called genes that code for different proteins. Now let's talk a little bit about a ribosome for a second. So a ribosome is located in the cytoplasm. But the thing about a ribosome is that a ribosome itself is made up of protein. So let's just go here. So it's a protein. It's a large protein, and it's actually wrapped by RNA. So it's like a sort of like cardboard wrapped by string. And so this structure is the reason that it's actually called a ribosome, or the reason that RNA is around it is called a ribosome, because ribo stands for ribonucleic acid body. So this particular RNA isn't the messenger RNA that codes for a protein. I'll talk about this in detail later, but it's actually structural. It's something called rRNA, for ribosomal RNA, or pirates like to call this rRNA. So what's interesting is the ribosome itself. Okay, so there it is. There's a ribosome. And so let me illustrate what we had talked about before. RNA, messenger RNA is produced. Messenger RNA comes out over here. And it contains the recipe on how to make a protein. And so let's say that the protein is made up of amino acids. And so these amino acids come off of the ribosome and they make a protein. And you're like, okay, there we go, there's a protein. But what's somewhat clever about this whole process is sometimes the protein, I'm glad that worked. I've never done that before. Some of the proteins that are produced by a ribosome, this is the part that's a little weird, is that some of the proteins produced by a ribosome are proteins that are going to make up future ribosomes. So there's a gene that codes for a protein that will make up a future ribosome. You're like, whoa. So where is that protein going to go? That protein is going to travel back into the nucleus right here. This is a ribosomal protein, although I'm using the same color as DNA, so I'm going to like shade it in for a protein. And so in the nucleus, some RNA is going to be produced, but it's not messenger, it's ribosomal. So it's rRNA. And then that coils around. And look what I have made. I have made a ribosome. So a ribosome is made in part in the cytoplasm on already existing ribosomes, and then it's finished in the nucleus when rRNA coils around it to form a completed ribosome. So my point being, if you remember the picture of a cell where all these ribosomes look like pepper all over, what would happen if you sort of unscrewed your pepper container and just piled a big dump of it? It would look like a big black spot. And so in the nucleus, the area in the nucleus where there's a bunch of ribosomes being put together is called the nucleolus. So the nucleolus is in part a place where ribosomes are manufactured, because some of it is manufactured over here as well. And so Let's see if I can come out of this now. Go back here, go like that, and let's continue this discussion here. So in the nucleus, ribosomal RNA is being produced, and it's being coiled around proteins that were made in the cytoplasm, which are called ribosomal subunits because they don't make up the completed ribosome. So these subunits of protein pass from the cytoplasm back into the nucleus, where they're combined to form ribosomes in these darkened structures called the nucleolus. And so here's another illustration of this. Here's DNA. You make a small piece of messenger RNA that travels out of the nuclear pore, goes to an already existing ribosome. It makes a protein. So ribosomes are where proteins are made. So what's interesting is the ribosome itself, though it looks like turkey or a piece of chicken or something, itself is made up of these little pearls of amino acids wrapped by our RNA. And so the pearls themselves go back into the nucleus, and they're wrapped up by our RNA, and then a ribosome is produced. So this dark structure, I know that's a little tricky, that dark structure in the nucleus, so this is just the nucleus. Here's the whole cell out here. This happens to be a white blood cell. It's a red blood cell here. So white blood cell, this dark structure, is called the nucleolus, and it's where ribosomes are put together. Come back over to here. Let's go here. Whoops. It's not going to let me do that. That's okay. It doesn't want me to do that. Maybe if I did this, it will. Okay. So inside here are where the ribosomes are put together. So there's millions of ribosomes being formed, and then all these little black dots out here are ribosomes. Okay, so that's what I wanted to say about that. Here's another cool picture of the nucleus. It's not the cell, it's just the nucleus. Here's the nucleolus. These arrows right here are pointing to the nuclear pores. All these little black dots out there are ribosomes. And so ribosomes direct protein synthesis. And the way they're able to do that is they get the message from RNA, which contains the amino acid sequence, and that travels out into the cytoplasm where proteins are put together. And so basically the ribosome, again that looks a little bit like a turkey, is the place where proteins are manufactured. And some of the ribosomes are free, meaning they're just found in the cytoplasm, and some of them are actually connected to this internal membrane system that we're going to talk about in our next video called the endoplasmic reticulum. And that's pretty cool. And here's a picture enhanced. This is the transmission electron microscope. The ribosomes are shown here in red. The endoplasmic reticulum is shown here in sort of yellow inside. This is the nuclear membrane over here. This is a mitochondria. And so it's kind of cool. Free ribosomes are suspended in the cytoplasm, and bound ribosomes are attached to endoplasmic reticulum, but both produce proteins for the cell. And that's all I wanted to say about the nucleus and ribosomes. Hope you enjoyed that.